There are different models about how life achieved its current diversity. And in science, it's appropriate to consider the predictions of these models and to test them. Those of the evolutionary model, where humans would be similar to other organisms, such as other vertebrates, that there would be a pattern of similarities, and levels of complexity intermediate between very simple animals and humans. And this would be different from the creation model, in which humans are 100% unrelated to all other organisms and equally unrelated, so that there would certainly be no predicted similarities or pattern of similarities. And the intelligent design model, in which complexity is irreducible, and there would not be any organisms with some but not all of a complex feature found in humans. Well, if we were to consider genetic tests of these models, say for example, using the protein insulin, it is clear that not only do other organisms have the hormone insulin, but that insulin-like proteins are even known in invertebrates. And in primitive chordates, there are hybrid molecules which have some of the characteristics of insulin and insulin-like peptides, seemingly transitional between the two. With regard to where insulin is produced, in the creation model, hormones could be produced from anywhere in the body, including non-homologous regions. But in all of the jawed vertebrates, the hormone insulin is produced from an endocrine portion of the pancreas known as the islets of Langerhans, as given here not only in the human but also then in the frog. In the simplest fish, the jawless fish, the endocrine cells making insulin may not yet be part of the pancreas and may be interspersed in the intestinal epithelia. And so the pancreas in humans and jawed vertebrates is a composite structure composed of both exocrine and endocrine tissue with two separate origins. And in jawless fish, there are examples of organisms which have some but not all of these features. There are other similarities of insulin molecules which wouldn't be expected if humans were unrelated to other organisms, such as the need to cleave a few amino acids from the precursor form of insulin before it is active. Finally, if one were to compare the sequences of insulin molecules across different organisms, one would not only identify similarities. For example, chimpanzees have insulin molecules whose amino acid sequences are 98.2% identical to those of humans, but also a pattern of similarities. So for example, all of the sequences, which are more than 95% identical to the human sequence, are those of the apes and old world monkeys, the catarrhine primates, just as would be predicted by the evolutionary model based on other anatomical and genetic studies. And that there is a varying degree of relationship when comparing insulin molecules of other organisms, such as that of the rat, which is 83% identical. So the similarity of insulin genes, their physiology and signaling mechanisms, and their sequences overwhelmingly support the predictions of the evolutionary model.